going to talk about Spark. And like Holly said, we want to give time for you to ask questions and make this as interactive as possible. I think there's a lot of value when parents ask questions because that brings up what you really want to understand better. So we're going to talk about, we're going to talk about genetic testing of autism and other developmental disabilities. We're going to describe the risk for autism recurrence, expand the understanding of the autism genetic mechanisms, explain the benefits of genetic testing in patients with autism and developmental disabilities, and describe the Spark research study. So let's start. What is autism? Autism is an umbrella term that brings patients that have different clinical presentations. It's very heterogeneous. The kids with autism can look very different one from another. It's a neurodevelopmental disorder. And the two big characteristics of autism across the spectrum are that the social interactions are impaired and there are restricted repetitive patterns of interest and behavior. Additionally, we know that autism results from early alterations in the brain development and the connections between neurons in the brain. Looks like a lot of the difficulties with autism are in the synapses or connections between neurons. At this time, we don't have a blood or a medical test that can tell us if somebody has autism or not. And the diagnosis must be made on the basis of behavioral observations. Just a few words about the diagnosis of autism. Ideally, when we do the diagnosis of autism, we want to get a solid history of the the developmental trajectory of patients and the symptoms of autism. And so there are these different tools that you can use to obtain the history. I personally obtain my own history when I evaluate patients. And we want to understand what's the developmental level or the learning abilities of a patient with autism to try to understand the symptoms in the context of their learning abilities or the developmental level at that time. And then finally, the gold standard now for the diagnosis of autism is the Autism Diagnostic Observation Schedule Evaluation, which is a set, an observation based on patients are asked to do a set of tasks that are standardized. There are 3 different types of ADOS, depending on there are 5 types of ADOS. There's one for toddlers, and the others are based on the language level of the child and the age. The number 4 is for adults. But the clinician who is trained makes observations of the patients in the specific tasks and takes notes about social abilities, communication, restricted repetitive patterns of interest and behavior, and puts those observations in an algorithm that gives a score. And with this score, the patient can, gets if it's in the autism spectrum or if it's outside of the autism spectrum. And then when they are in the autism spectrum, it gives you a level of severity of autism, if it's mild, moderate, or severe. But this observation is standardized. This is the gold standard, but it's not a perfect tool. You still need an experienced clinician interpreting these observations in the context of the developmental learning abilities and the symptoms that are described by the parents. So with all these things, we come up with a diagnosis, and then we clinicians do like a recommendation based on at different levels. So something that is important to be aware of is that, and this is encapsulated by Dr. Stephen Shore. He's a professor in New York. He's a professor of special education in New York, and he said, if you have met one person with autism, you have met one person with autism. And this tries to explain that there's so much heterogeneity in the presentation of autism. And the variables that are gonna make people look different is, you know, the learning abilities. There are some people that are very bright and have learning abilities that go beyond 2 standard deviations above the mean, that are gifted. And there are others that have learning difficulties, intellectual disabilities. And then you see patients that are in all this range. There's a variety of language abilities. The majority of patients with autism are late talkers, but there are many patients with autism that have good core language skills, that speak on time, and they have a nice variety of vocabulary in their language, good syntax, good grammar. So that can make them look different depending on what's the level of language. And the autism severity can also be mild, moderate, or severe. The role of the family, the motor skills, the presence of comorbid psychiatric conditions. Like many patients, 50 to 80% of patients are supposed to have ADHD. There are like up to 50% of patients that have anxiety, and many may have depression, or sometimes they can have bipolar disorder. And so these conditions are gonna make patients with autism look different. And the trajectories, there are some children that are diagnosed early on, and they can look very similar, but over time, some of them may have a really positive trajectory and make a lot of quick progress, and others don't make as quick of progress. So there's some intrinsic trajectory that sometimes patients bring. Ideally, we recommend intense services for everybody, because we believe that providing intensive services is going to improve their trajectory, whatever their trajectory is supposed to be, and it can make it better. And we feel that, you know, we want our patients to get as many services as possible and to have the best trajectory possible. But it's hard when we make a diagnosis for a young child. It's hard to predict how are they going to do. We sometimes speak on some characteristics that tell us if the trajectory is going to be more favorable or not. But still, you know, you have to see how they do over time. So something that we talk all the time is that autism is not a final medical diagnosis. Autism is a behavioral diagnosis that tries to describe, encapsulate social deficits and restricted repetitive patterns of interest and behavior. But when you think about diagnosis, you want to find the cause. And just saying autism doesn't tell you the cause. So we're not done with diagnosing autism. We want to know more about what caused their autism and try to understand that better. So we do order genetic testing. And this is the focus of this presentation today. So autism is considered a primarily genetic condition, but there are also environmental factors, as we're gonna say. The SPARK study that has more than 30,000 trios at this time, more than 30,000 participants, found that there's a concordance rate of 77% for identical twins, meaning if you have 100 identical twins, 77 are both going to have autism, and in 23 cases, one of them is going to have autism and not the other. So this points towards like a very genetic condition, but it's not purely genetic. Otherwise, it will be 100% the concordance rate. In fraternal twins, the concordance rate is not as high as 31%, and that makes sense because they don't share they have variability in their genetic makeup. And siblings, the concordance rate in the SPARK study has been 20%. And these are the results from SPARK are consistent with a number of landmark studies that were done with twins in the 90s. There were over 30 twin studies, and they all coincide that there's a high concordance rate with identical twins and a little bit less in fraternal twins and siblings, like we're showing now. So how much of autism is genetic? So the estimation is that 50 to 80% of autism is genetic. And in a large population study in Sweden, there was a concordance rate of 80%. There are also environmental factors, but our understanding of environmental factors is still limited. We're hoping to learn more. There are ongoing studies to investigate more those environmental factors, but we don't know them that much. So we know that there are some medications that can increase the risk for autism when pregnant moms take them. Folic acid, the lack of folic acid during pregnancy may be a risk factor. The mother having some medical problems. And some people talk about pollution from cars could be also a factor, but we don't know the extent on how much, and looks like there's a lot to learn. So hopefully in the next few years, looking at those studies results, we'll be able to have a better sense of that. So, so we're talking about 50 to 80%, maybe closer to 80% genetic causality of autism. But yet, when we order day to day, we order genetic tests, the yield of these genetic tests is only 10 to 20%. So what happens with the others? We don't understand very well. The other fact that is very interesting is there's no single genetic cause of autism that accounts for more than 2% of autism cases. So there are like 400 genes that have been described to cause autism, but none of them goes more than 2%. The majority are under 1%. The one that goes closer to 2% is the Fragile X. So, so when we look at the kids that have a diagnosis of autism, we find that in 20% of cases, they have features like the eyes are a little more separated, their ears are kind of low set, their hands, they may have a finger that is a little deviated, they have some skin lesions. So we call those, you know, there are some signs in the physical exam that point that there might be an underlying genetic condition. So in those cases, we call syndromic when kids have some of these morphological features that suggest a genetic condition. But the majority of patients with autism don't have those morphological differences. And you cannot tell just by looking at them, but in 20%, you can tell that maybe there's something there. And it's interesting that the yield of genetic testing in those that look a little bit different, the yield is 50% for finding, up to 50% for finding a genetic cause. But the yield of doing genetic testing in a kid that doesn't have those features is much lower. So it's important to notice that. So what are genes? What are chromosomes? So if we look at a gene is made up of DNA, and they provide instructions for making proteins. So each gene is going to be coding for a protein. And these are important, many times they are important proteins that have an important role in the functioning of our systems. So, so the gene is the really important unit that is going to code for the proteins. But then we have the chromosomes. The chromosomes, the genes are located in the chromosomes. And everybody has one set of chromosomes from the mother and one from the father. We have 23 chromosomes from the mother and 23 from the father. And the genes are in the chromosomes, and they're gonna be coding for the proteins. So, so here's a gene that is unaffected and is coding for a protein, and the protein is working and is doing its job. But what we are looking is for genetic variants. So when there's a genetic variant that is significant, there's something in the code that is not going to be quite right, And so the protein that is produced is going to be non-working, or it's just going to be missing. So those are the genetic variants that we're looking for that could explain some cases of autism. So interestingly, things are not black or white. You have some genetic variants that are very significant, that are coding for critical proteins that if the individual doesn't have that protein, that may lead to autism or a significant neurodevelopmental disorder. But there are other genetic variants that code for proteins that are not really important, that don't have a key role, and that there might be other genes that code for the same protein or for a similar protein. And then you have genetic variants that go in these spectrums from significant to benign, that may cause more or less difficulties in the individual that carries them. So something I wanted to mention is, a lot of people don't know that we have 3 million genetic variants in our genetic makeup and our genetic code. And these genetic variants are classified as common variants that 95% of our genetic variants are shared with 5% of the population, so they are considered common. And there are some rare variants that are less than 1% that are rare or unique to one individual and their family. So those are considered rare variants. So an important concept that I think we need to share here is the de novo genetic changes. When we talk about genetic, everybody thinks about, okay, if this is genetic, that means that either dad or mom passed the genetic variant to the child. But there are some situations where the genetic variant is not passed by mom or dad. There is something that happens during conception, and when the sperm fertilizes the egg, there's a genetic change, and there's a genetic variant that happened during fertilization. And that's what is called a de novo variant, a change that is not carried by father or mother. And something that has been found is that in autism, these de novo genetic variants account for 20 to 30% of the cases of autism. So this is a very important concept to keep in mind. And I think this correlates with the fact that sometimes when I talk to families and we break the news, many times parents ask, where does this come from? Because we don't have any history in the family of autism. And this could explain that it was a new genetic variant. So let me talk a little more about the common genetic variants and that can have a role in the causation of, in the causality of autism. So these are relatively high frequency genes that each one of these highly frequent genes can give a little bit of risk for autism, but it's low. So just one of these high frequency genetic variant would not cause autism. You would need up to 5 hits of low risk genes to cause autism. And so this is another case where there might not be a history of autism in the family, but somehow maybe dad has a couple of these common variants that give a little bit of risk for autism, and mom has 3 of those. And then when they get together, they may have one child that has the 5 and develops autism. And there are the rare variants that I was mentioning before. And it looks like for autism, there are a few rare genetic variants that give a lot of risk for autism. And just having one of those genetic variants may cause autism. And a lot of those may be like de novo changes. Once in a while, there can be passed by the parents, and somehow it didn't cause any trouble in the parents, but it did in the child. So when we think about the transmission of autism, we need to think that sometimes it's just one gene that is different. But sometimes it can be a contiguous gene problem, like this a chunk of genes, a piece of material, genetic material that is different, that there's a variant that is missing or that is duplicated. And sometimes you have what we discussed, like it's a combination. There are these common genetic variants with some environmental factors that lead to autism. And it looks like these are the majority of cases. So I want to talk now about a very nice model that attempts to explain the complex genetic transmission of autism. So this is, I want you to think about this cup. This is a cup, and there are these balls. There's one, the green one illustrates the common variants, the weaker genetic factors. The purple one is the rare variants, and these balls are longer, and they give more risk for autism. In some cases, just one of these may cause autism. And then the orange balls are the environmental factors, which we don't understand very well, but we put them there because we know that they are important factors. So here we have an individual that has 3 environmental factors and 2 common variants with weak genetic risk for autism. And this individual doesn't have autism. And this other individual has 3 of the common variants, 3 of the environmental factors, and 1 of the rare variants that give a lot of risk for autism. So this individual has autism. Let's look now at the family. There's a father and a mother. They both don't have autism. And the father has just one common variant with a small risk, weak risk for autism. And then we have mom that has the same one risk factor, one common variant that gives weak risk for autism, and 1 environmental factor. But then mom has this A gene, which is a rare gene that gives more than the common variants, gives more risk. So she's kind of a carrier of that, but it's not big enough that she got autism, but she's carrying this risk for autism. So they have children, and child 1 got mom's rare variant with high risk for autism, and then she got the dad's, the mom, and 1 de novo variant that give small risk for autism. That are common variants, and he also has some environmental factors. So this individual, unlike mom, this individual has autism because in addition to this medium-sized rare variant, medium-sized risk factor genetic variant, has these other common variants that each one gives a small risk, but there are 3 hits plus the hit from the rare variant that makes this individual reach the threshold. Child 2 doesn't get the risk factor from mom and has just 1 common variant that gives a little bit of risk for autism. So this individual doesn't have autism. But then look at child 3. Child 3 gets a genetic makeup that is very similar to mom. So child 3 won't have autism because the risk didn't reach the threshold, but child 3 is going to be a carrier of this variant. So if you think about child 2 and child 3, child 2 is going to have low risk for having a child with autism, but child 3 is going to have relatively high risk for having a child with autism. And then finally, we see child 4 that has a big rare variant that gives a big risk for autism and just has 1 common variant, but this rare variant is so big, has so much risk for autism that this individual develops autism. But this is a de novo variant. So this is not something that came from parents. And probably child 4 is going to look very different than child 1 because they have their big risk factor for autism is going to be different. So this table tries to illustrate the fact that females in autism behave different than males as a group. Because there are some females that have similar presentation than males, but most females tend to have a milder presentation of autism, and it's a little harder to identify sometimes when females have autism because they somehow compensate better. The way they kind of try to explain this, they say that females have a bigger cup than males, so they need more genetic risk factors to develop autism. And finally, this is a very busy illustration, but I want you to focus on individual one. We're just going to talk about individual one, and they are talking about this gene, ASTN2 gene, and how this gene is a rare variant that gives a lot of risk, not just for autism, but also for obsessive compulsive disorder and also for attention deficit hyperactivity disorder. So, and this is something important to keep in mind, that autism is not an isolated neurodevelopmental disorder. Like a lot of people have a combination because the genes that lead to autism also give risk for other conditions. Like the most common that we said is ADHD, anxiety, but also there's some shared risk with autism, with seizures. Some patients with autism can have seizures, and that is often because they got a gene that gives risk for these two conditions. So this is one of the reasons it's really important to try to identify the cause, because sometimes if you can identify the gene variant, you can kind of anticipate the trajectory that some individuals with autism are going to follow, and also you can anticipate the presence of other conditions like a seizure disorder. You cannot say if somebody's going to have a seizure disorder, but you can at least have an idea if they have more risk for having a seizure disorder or other conditions. So that brings me why to do diagnostic genetic testing. First one is diagnostic clarity. I think people relate better when there is when you can find a genetic cause for the condition. You can understand where did that come from, and then you can compare how your child may do with other individuals that have that condition. It can help if you compare with other individuals with the condition. You can anticipate or predict the developmental trajectories. It can also help to understand the risk for the parents to have other children with autism, but it can also help understand the risk for the siblings or the immediate family for having offspring with autism. There are some genes that are associated with some medical morbidities, medical conditions, so we can anticipate that. And it also, having a genetic diagnosis sometimes can help allocation of supports and services. So these are a couple of examples of conditions that having like this genetic variant has been associated with cardiac defects. So you need to look for cardiac conditions. This one is associated with diabetes, and the other is associated with obesity. So something I want you to see is the scale of genomic variation. So I mentioned there are tests that look at the chromosomes, and you can diagnose Down syndrome or these big chromosome conditions with these tests. But for diagnosing the causality of autism, this wouldn't be enough. So you really need to do a test that looks at this level to identify genetic variants. That's the test that we use now. The standard test to look for this is the microarray. And but there are some conditions like Fragile X that is just one gene. It can be Fragile X, it can be Angelman syndrome. So there are tuberous sclerosis, and those conditions are just one gene. So the standard test, the microarray, won't be able to pick such a small of genetic variant. So you really need a test that looks specifically for that condition to be able to pick that up. The clinician needs to suspect it and needs to target that specific condition. So there are a number of conditions that are overlooked with this, and for this you need something more specific. So the microarray is the standard tool that we use. That's a standard test that we order. And it looks to a great number of genetic variants that have been associated with genetic conditions. It's not all inclusive. It just looks to a lot of these genetic variants that can have a role in causing problems. The test that is coming up, that is already with us now, is the whole exome sequencing. And the exome sequencing is a test that has the potential to look at the whole genome. So you are sequencing the whole genome. And the exome sequencing looks at the functional parts of the genome. And at this time, it doesn't fare that much better than the microarray, but it has the potential to be much better down the road. It's going to look at the spelling, and it potentially could pick up some of these monogenetic, like one gene conditions, but it's not as great on that yet. So what kind of results do you get? I mentioned that the yield to find something is 10 to 20%. So sometimes you get a microarray or a whole exome sequencing and it says normal. We didn't find anything. So what does normal mean in a genetic test? It means that, they didn't find it. It doesn't mean that this child doesn't have a genetic variant behind their autism. It means that they couldn't see it with the technology that we have at this time. But maybe down the road, with better technology, we'll be able to pick up. But at this time, we can't. There are other test results that are very clear. Okay, we found this, and this is clearly associated with autism. The literature shows that kids that have this condition have autism. Okay, you have a clear answer. Then sometimes you find something, but then when they do the search for what does that mean, they realize that it's not significant because they know that those areas in the genetic makeup are not relevant. So you just dismiss that. The problem is that we get a lot of these results, which is called a variant of uncertain significance. So we found something, but we don't know what does that mean because it's not being described in the literature. And that can be frustrated for parents. And so what we do in these cases is we ask for parents we test parents, and then we try to interpret that based on parental testing. If both of parents don't have it, it makes it likely that this is a new variant, like a de novo variant, and it's probably significant. But if parents have it, then it requires more analysis, and it becomes a little more complicated. So what's the yield? Like I'm showing here studies that show that the yield is 8, 9, 10%. If you have kids that look like they have different features that suggest a genetic condition, the yield is much better. Okay. So I have, do we have any questions before we go to talk about the SPARK study?
Yes, so I think a few questions came in, and some of them really linked to percentages and likelihood when it comes to autism risks related to siblings, as well as, well, actually, why don't we start there? I think you did mention this when you talked about some of the studies that SPARK had put out on identical twins, as well as fraternal twins and siblings, but do you want to just reiterate the likelihood, if a family already has a child with autism, what's the likelihood of potentially having another child with autism?
So it's hard to say a straight number if you have one child with autism. So there's a big range. So people talk about 2 to 20% of risk for having a second child with autism. If you do the genetic testing and you find the cause, you may be able to narrow that number. Probably would be in the lower end if this is a new genetic variant that caused this child's autism. The number for having a second child with autism would be low. But if you find a cause that you need to analyze if parents passed it to them, so there can be a little more variability. I would encourage, if parents are interested in having a second child but want to understand well the risk, I encourage them to go through genetic testing to try and then consult. I personally order genetic testing for my patients, and most of the problem is that the yield is not that great. The yield is 10 to 20%. So a lot of patients, the majority of patients, don't have an answer. So that doesn't make a difference in this big range that I'm giving, the 2 to 20%. But
right. And we do know that risk increases when multiple siblings have a diagnosis of autism. Then
having 2 children with autism, that increases to like 40 to 50% having a third child with autism. And we definitely know families that have 3 children with autism, but it's not common, but it's something that can happen.
And then we had a question, and I'm not sure there are studies on this. I know there have been studies on twins. Have there been any research or studies done on the chances of having triplets with all 3 having autism?
I have a patient, triplets. I think there would be a higher risk of having the 3 triplets with autism if they come from one egg. So they have if they are identical twins. Mm-hmm. But if they are not identical, then the risk could be more like fraternal siblings or fraternal twins. Mm-hmm. There will be a similar risk. It's possible, depending if they are all sharing this. In this case, there would probably be a rare high-risk genetic variant that is shared by the 3 of them. But I think it's possible.
Yeah. And I think on that note, we had a question about how we know whether or not any environmental factors exist at all. And I think we gleaned some of that from that identical twin study. So do you want to take that question?
Yeah, that's the understanding, because we have the identical twin studies that talk about a risk of 77 to 90% in 30 studies done. So that very clearly tells you that it's a strong genetic risk, but it's not 100% genetic, because there are some situations that one of the identical twin has it and the other doesn't.
And if you have individuals who present differently, so obviously autism is a spectrum ranging from individuals who are more severely impacted to those that have high verbal communication and might be a little more independent in their living skills. When it comes to individuals who might be more independent or labeled as high functioning, does that change any of the impacts on chances for having siblings with autism?
Not that I know. I think I don't think it's gonna be less risk. I have actually seen situations where there's one of the children that have a higher form of higher functioning, milder autism, and another child that has that are more impacted and have more developmental delays. So I think we don't understand well. I like the concept of the cups, the explanation, because it tells you how the different genes can interact to give risk. So so I would say not necessarily. As far as I know, it shouldn't be less risk because it's a milder, higher functioning form.
Right, and a lot of that, it sounds like it depends on the types of risk factors and the types of genetic variants that the parents may have. And so it sounds like genetic testing in those situations could certainly be helpful.
Yeah, but
before we move,
would be helpful, but may not give you the answer necessarily. But you know, I personally think it's good to leave that stone to see if we can get more information.
Absolutely. Before we move on to Spark and the type of genetic testing that can be done and what we've learned through Spark, one more question. If we have a normal microarray, would the Spark study be able to find any different information? And so this speaks to, for families who have already done genetic testing, why might they still, or is there a benefit to still participating in Spark?
Should I answer that now or after going through the Spark presentation?
I guess yeah, that's a good point. Why don't you go through Spark and maybe answer it while you're chatting about Spark?
Yeah, that's a great question for the Spark presentation. So what is scientific research and what is a Spark? So Spark stands for Simons Foundation, which is the sponsor for this study, powering autism research. The mission is simple, is speed up the research and advance our understanding of autism to help improve lives. So like I mentioned before, the genetics of autism is so complex. We need to have a better understanding. Something that we know from other neurodevelopmental conditions where we understand the genetics is that when you understand the genetics of a condition, you can develop animal models for that condition, and you can start experimenting and understanding better what happens, how is that genetic variant lead to the changes in the brain that lead to, like in this case, autism. And once you understand the mechanisms, you can try to come up with treatments, and you can try those treatments in the animal models, and then eventually come up with a targeted brain treatment for humans. So I want you to make the connection that an understanding of the genetics of autism will eventually, we're not there yet, but will eventually lead to the development of treatments that can help treat this condition. So that's what they mean with speed up research and advance our understanding of autism to help improve lives. So families enrolled in this study, they don't need to come to Rush or other center that does Spark. They can just do their enrollment online, and they will get some kits to collect saliva samples that will be mailed back for analysis for the genetic testing. And this is the link for the study. So the goal of this study, there are like, the last time I heard, there were 26 centers across the country. Rush is the Illinois site, and trying to recruit 50,000 families with autism, collecting data and genetic information. The goal is to return genetic results so participants can be grouped by genetic similarities or phenotypic traits. And eventually the goal is to match participants to research studies. So eventually, you know, when you participate in Spark, you are helping improve the understanding of autism, which eventually will lead to an acceleration of the access to treatments. You are also becoming part of a research community, a nationwide research community, where there are a lot of autism researchers. And eventually you can have opportunities to participate in some studies, which you may always say, you may say, thank you, but I am not interested in this study. but you will have the information about these studies, and then at some point you may feel like you want to try some study. So Spark is not looking at this time for a cure for autism, but the idea is that it's going to help the understanding of the genetics of autism. It's going to help the development of treatments. At this time, like the yield for getting a result, a genetic result, is up to 10%. They are not doing genetic tests for autism. So who is eligible? Anybody that lives in the United States that communicates in English, and it can be an adult with autism that lives independently, or there can be parents of a child or dependent adult with autism, and biological siblings are welcome. There's a need for a professional diagnosis of autism.
And then one correction, I'm so sorry, Cesar, one correction, or not correction, but one addition come January 2022. So in just a few weeks, that number 2 will be participants should be able to read, write, and understand English or Spanish. So both of those are.
Thank you, Holly. So how is the study? Here is a diagram that tries to show you. You register online, and online you give the consent to share data. You enter basic information about the family structure, and then you consent for the DNA analysis. And then you have to invite, this is one of the parents that registers him or herself and the child. And then they invite the other parent, and the other parent needs to access this site independently and give the consent. And then the kids come via mail, the kids to collect the saliva. Sometimes it can be a little more challenging to collect the saliva from the individual with autism, but there's like a swab with a video that can show you how to do that. And then you're asked also to complete some surveys. So eventually they're gonna do the DNA sequencing, and they're going to return results when they are available. Something that is really neat about this study is that even if you don't have a result from the first run of saliva, they're going to keep running this sample every year. And as the number of genes discovered to cause autism expands, so it may take a little bit of time to get an answer, but you may get an answer at the end. So what do we know now from the 6 years of Spark? So we have like 24 samples from individuals with autism and their family members. We have returned 10% of genetic results. They usually report only the positive results. And genetic analysis has increased the number of genes. So scientists have discovered over 200 genes. The scientists predict that several hundred more genes related to autism have yet to be found. And finding the cause for autism, like we mentioned before, may contain important clues. So usually there's a committee that analyzes the data at Spark and determines when there's enough information to consider that a gene has the potential to cause autism. So this is what families get when they have a positive result. They get a report that explains the genetic variant and what we know about that genetic variant. So what is in there for families? You get the opportunity to join a national community for autism, and there are more than 43,000 people. It's going to help understand the enhanced knowledge about the causes of autism. It's gonna there's an Amazon gift card for participants. It's gonna help keep up with the new discoveries. You're gonna be communicating with the Spark study and receiving information about the the progress in the study. You may get a genetic diagnosis in the short or in the intermediate term, and when you get a result, you have a personalized report. So we do have an upcoming event. It's in Franklin Park, and it's going to be on Saturday, December 18th from 9 to 2. You're welcome to come, and we will help you enroll in the study, and we can help you collect the saliva sample from your child. And that's what I wanted to say, and I look forward to your questions.
Wonderful. Well, thank you. I am going to look through some of the questions. And just to add, along with getting those, if a family does have a genetic result, just a couple updates. So if they do have a genetic result, along with that information, they are also paired with a genetic counselor. If they already have a genetic counselor that they're working with, that or a physician they're working with, they can. Otherwise, Spark will provide that, because it's a lot of information and it can be really complicated in terms of understanding it and helping to identify what does that mean. It also connects families to a community. It's called Simon's Searchlight. So for those that do get a result, they're then connected with other families and networks and providers that are specific to that genetic result, which I think has been really impactful for families. So Cesar, do you want to speak a little bit to that difference between, for families who've already done genetic testing and done that microarray, would Spark be able to find any different information?
Yes, I didn't answer that question, and I was looking at that. The test that is done by Spark is not the microarray, which is the standard test. The test that is done by Spark is the whole exome sequencing. Is the last test that I mentioned in my presentation, which is a more advanced test than the standard test. At this time, the yield is only 10%, but there are some individuals that in whom we don't find anything in the microarray, and we may find something in the whole exome sequencing. So we always recommend that that you do both, because they can add to each other. And then the nice thing is that they are going to keep checking it every year. So even if there's not an answer right away, there may be an answer down the road. So we always encourage to do the standard test in addition to the participation in the Spark.
Absolutely. And then we had a question come in, just to be clear, who do you need saliva from, essentially? And we understand all family structures are different. And the qualifications to participate in Spark is that you have a professional diagnosis, and the individual with autism is submitting that sample. When possible, both one or both biological parents are also invited to participate, because when we have the sources of the DNA, so both biological parents, we can learn a little bit more specifically related to inherited versus de novo traits. And so a sibling or a half sibling is also welcome to participate, up for each family, up to one. It doesn't replace the biological parents, but I do want to emphasize having both biological parents is not a qualification for the study. Really, you can have just the individual with autism submit theirs, and that would they'd still be able to participate. It's just when possible, it can give us more information. We had a question come in. What is the strongest researched environmental factor thus far? Or can you speak a little bit to environmental factors?
Yes, like I mentioned before, there's not a lot on environmental factors, but the strongest environmental factors are the ones that are associated with pregnancy. So that, like it was mentioned during the presentation, there are some medications. There can be some exposure to drugs in utero or alcohol have been associated with increased risk for autism. And then also prematurity appears to increase a little bit, not a lot, but it increases a little bit. Extreme prematurity can increase a little bit the risk for autism, and other complications that mom may have that cause an insult to the fetus brain that can also increase a little bit the risk for autism.
And then we had a question, and I'm happy to take this one, on the time commitment to participate, how long it takes for surveys, and then about security. So in terms of the time commitment to participate, registration, the online portion, takes about 20 minutes. The saliva collection honestly depends how much saliva you have in your mouth and how easy it is to get it from your child or adult. That can really range from less than a minute to, a good 20-minute process. But we say generally the registration process takes about 20 to 30 minutes with that online registration and saliva collection. There are surveys that are optional. I think families get an additional Amazon gift card after filling some of them out for their time, but the surveys online take anywhere between 10 minutes to over an hour, and so those are really optional. What they do is they help us get additional background information, particularly when a genetic result is identified. Having that additional information and those factors can be really helpful. When it comes to security of genetic information and who has access to it, it's a great question. I think both with data and privacy, as well as with genetic information, all families, when they submit a sample, there is a barcode associated with it, so that when the lab receives the samples, the family's or the individual's name is taken off, so that information is de-identified and only provided to qualified researchers, meaning only provided to teams and to Spark. It will never be shared with any third parties, especially without consent. When it comes to data in that research match, we never share family data or family information with other researchers. What we do is for researchers that qualify, and it might be a match for families, what we do is we send families the information of, are you interested in this study? And only then would once you are, you confirm interest and indicate that you would like to have your information shared with other researchers, will that be done? So the answer to that is never shared, and only the Spark team members will have access to that. And then when your samples go to the lab, they are de-identified. In terms of information for the 18th, we can definitely follow up with that or you can look out for that in your emails. They will come through as a follow-up to this webinar, so you will get information on that. And then we had another question come in. We had a family that participated, and they wanted to know how close they are to getting results. And so, Cesar, do you want to speak broadly a little bit more to how long it takes to receive results? Because this has definitely changed over time, but it is not quick. So do you want to speak to that a little bit?
Yep. So, yeah, it takes a little bit of time to get the samples processed and to get a result when it's positive. The thing is, they will only tell you if they will only report the positives. So you will be contacted for results only if the testing on your child was positive. So what we can do is we can take your information.
Cesar, I'm so sorry. That's actually a new change. The study is now on an annual basis going to be sending out when families do not receive a result, or when there's not yet a result. They will receive that notification now.
Oh, okay.
Yeah.
That's great. That's great. But this family has not received any, sounds like they haven't received any information yet since 2019. So hopefully this year you will get a report telling you what's going on with the sample.
And if not, what I would recommend is contacting that spark@rush.edu, because we can look into that and have our coordinators look into that for you. That's when families apply, making sure that they have updated email and phone numbers in there so that the Spark team can get in touch. And if you feel like that time has passed and you're wanting an update, that's what we're here for. So we're happy to answer those questions and kind of check in on the status of any of those results as well. Any other questions? I realize we are now about 2 minutes over, so I think we were able to get through all the questions. And so unless there are any burning or lingering questions, I think this would probably be a good stopping point for us. Right. Well, I want to thank, as one of our participants said, thank you for all you do, Dr. Ochoa. And thank you to everyone who was able to join us and for your wonderful questions. Do feel free to reach out to us if you have any questions. We'll follow up with information about the event for those that are interested, and we are here to answer your questions if you have them. Okay.
Thank you.